There's a blood type so rare that in certain cities across Asia, you could walk through 10 million people and never once find another person who shares it. It shows up on hospital charts and donor cards as 2 letters and a small minus sign, type B negative. It looks like nothing, just a clerical detail, a medical footnote, the kind of thing you note down once and never think about again. But hidden inside those 2 letters is one of the most remarkable genetic stories in human prehistory, a story that crosses frozen grasslands and ancient mountain passes. It involves 2 completely different peoples, separated by thousands of miles and thousands of years, who should statistically have almost nothing in common, yet somehow left their fingerprints in the exacto same set of genes. Scientists spent most of the 20th century unable to tell this story, not because the evidence was absent, but because the tools to read it did not yet exist. Those tools exist now, and what they have revealed changes the way we understand not just a blood type, but the movement of human beings across the entire Eurasian continent. If you carry B negative blood, you're carrying a genetic signature that most people on this planet will never possess. You're holding in the cells of your body the compressed record of migrations that happened before writing, before cities, before anything we would recognize as civilization. Today we are going to follow that signature all the way back to where it began, both of its beginnings, because this story starts in 2 completely different places, and the fact that it ends inside a single person is the most extraordinary part of all. Start with the numbers, because the numbers alone should stop you. Human blood comes in 4 main types, A, B, AB, and O, layered on top. Of that is the Rh factor, which is either positive or negative. Combine those 2 systems, and you get the 8 blood types that appear on medical charts around the world, but they are not spread evenly across the human population. The distribution of blood types across the planet is one of the strangest and most revealing patterns in human genetics, and it has puzzled researchers since blood typing was first developed. Type O dominates the Americas. Type A is common across Europe and parts of East Asia. Type B, the one that concerns us today, has always been the anomaly. It is rare in Western Europe, falling to single digits in countries like Norway, Portugal, and the Netherlands. It was essentially absent from the indigenous peoples of the Americas and Australia before outside contact, but move east into Central Asia, northern India, Mongolia, and the ancient corridors of the Silky Road, and type B is suddenly everywhere. In some regions, it reaches more than 1/3 of the population. It is as if the blood type drew its own map across the surface of the earth, a map that ignores modern borders and coastlines and follows something far older. Instead, now add the negative factor and the puzzle becomes something close to a paradox. Across most of Asia, Rh negative blood is extraordinarily uncommon. In China, Japan, and much of East Asia, fewer than 1 person in 100 carries it. In some specific communities, it barely registers at all, a statistical ghost that appears so rarely in donor databases that hospitals sometimes go months without seeing a matching sample. The Rh negative factor, as we will explore in detail, is a marker associated with a completely different part of the world when you trace its history, a completely different population, and a completely different chapter of human history. So when you take a blood type that peaks in Asia and fuse it with a factor that almost completely disappears in Asia, you get something that should barely exist. A person who is B negative in East Asia is carrying 2 traits that statistically have no business appearing together in the same body. One pointing east, one pointing west, both ancient, both rare, and together creating a blood type so uncommon that in a city of 10 million people, you might be alone. This is not a small thing. In parts of China and Japan, B negative blood occurs in fewer than 1 person out of every 200 to 300 individuals. In emergency medical situations, finding a compatible donor can be genuinely desperate. Hospitals maintain watch lists, and rare blood registries track known B negative donors across entire regions. A single compatible donation can be the difference between life and death in a way that is simply not true for more common types. The rarity is not theoretical. It is immediate, practical, and for some people, it has been a matter of survival. But the medical reality is only the surface. Beneath it is the deeper question that geneticists spent decades trying to answer. How did this happen? How did 2 markers from opposite ends of a continent end up together in the same bloodline? To follow that answer, we have to go back much further than most people expect. The starting point, oddly enough, is a laboratory in Vienna in 1901. A physician named Karl Landsteiner had noticed something that had baffled doctors for centuries. When blood from one person was mixed with blood from another, it sometimes clumped into a dense, lethal mass. Other times it blended perfectly. Transfusions were a gamble. Patients died from what should have been life-saving procedures, and nobody could explain why. Landsteiner began systematically mixing blood samples from his own colleagues, testing every possible combination, and slowly a pattern emerged. There were categories, there were types. He had discovered the blood group system, the reason some blood mixes safely while some kills, and it would eventually earn him a Nobel Prize. But Landsteiner had only opened the door. He could sort people into groups, but he could not tell anyone where those groups had come from, why type B existed in some populations and had never appeared in others, or what the pattern of blood type distribution across the world actually meant. Those questions sat unanswered for almost 100 years. It took the development of population genetics and eventually the ability to extract and sequence ancient DNA directly from skeletal remains before anyone could follow type B back to its origin. When they finally did, the trail led somewhere that surprised almost everyone. When scientists began testing the DNA of ancient Europeans, the people who had inhabited Europe before recorded history, before the Roman Empire, before the Bronze Age, they found something striking. The specific segment of genetic code that produces type B blood was absent. Not rare, absent. It was not present in the early hunter-gatherers of Europe. It was not present in the first Anatolian farmers who arrived to introduce agriculture to the continent. Type B simply did not exist in Europe's original populations, and then, in the Bronze Age, it appeared. It arrived from the east, carried by people who came sweeping out of the vast grasslands of Central Asia. The blood type did not evolve slowly in place. It arrived the way travelers arrive, moving along a road that already existed, carried by people who were already in motion. That road was the Eurasian steppe belt, the enormous band of open grasslands stretching from Central Europe across Russia, Kazakhstan, Mongolia, and into the heart of Asia. For thousands of years, this landscape was the domain of nomadic herders, peoples who did not build permanent cities, did not draw fixed borders, and did not stay in one place, long enough to be contained by geography. They followed their animals across the open land. They traded, raided, and mixed with every settled population they encountered at the edges of the steppe, and wherever they traveled, they carried their genes with them, including the genetic code for type B blood. This is why type B does not radiate cleanly outward from a single point of origin, the way you might expect a trait to spread. It does not follow a simple expanding circle from one homeland. Instead, it traces the roots of people who were perpetually in motion, people who moved back and forth across an enormous geographic range across many centuries, mixing with populations in every direction. The distribution of type B blood across the modern world is not a map of one migration. It is a record of movement itself, repeated thousands of times across a span of time almost impossible to fully grasp. None of this movement would have been possible at the scale it achieved without one animal, the horse. When the peoples of the steppe learned to ride and later to harness horses to wagons, they gained something that no settled farming society could match: range, mobility, the ability to cover in days what took others weeks on foot, to move entire communities and their herds across distances that had previously been impassable barriers. Populations that might never have encountered each other suddenly shared bloodlines. Communities separated by hundreds of miles became connected through trade, marriage, and an occasional conflict. The horse transformed the steppe from a barrier into a highway, and type B blood was one of the passengers that traveled down it generation after generation, reaching toward every horizon. Look at where type B is most common in the world today, and you are essentially reading a map of ancient movement. Northern India, where Indo-Aryan migrations deposited their genetic inheritance. Mongolia, the heartland of steppe herding culture. Siberia, where populations moved along the northern corridors of the continent. And the corridors of the Silk Road, where the nomadic world collided repeatedly with the settled world of China, Persia, India, and eventually Rome. These are the places where the ancient steppe world and its genes left the deepest impression. Type B blood rode along with every one of those contacts, passed into children who passed it to their children, spreading not through the single dramatic conquest of one enormous empire, but through countless small human encounters repeated across a timescale so vast it is almost beyond comprehension. This is what genetics has made visible that written history never could. Written records only reach back so far, and they were kept by a tiny fraction of the people who actually lived. The vast majority of human beings who ever moved, settled, traded, and mixed across the Eurasian landmass left no document behind. No inscription, no memoir, no account of who they were, where they came from, or whom they married. They are invisible to history, but they are not invisible to genetics. Genes do not care whether you were a king or a herder, a merchant, or a migrant with no name anyone ever recorded. They log every contact, every mixing event, and every movement of a population, preserving that record in the bodies of descendants living thousands of years later. The spread of type B blood across Central Asia and into the edges of Europe is one of the clearest signals in the ancient human genetic record. It is a fingerprint left by people whose names were never written, still circulating in the bodies of their descendants today. But type B is only half of the story. The second half begins on the opposite end of the Eurasian world entirely. To understand the negative part of B negative blood, we have to leave the open grasslands of Central Asia and travel to the far western edge of Europe, to a mountain range straddling the border between northern Spain and southwestern France, to a people whose story is in many ways as strange as the story of the steppe nomads, though stranger in a different direction. The Rh factor gets its name from an unlikely source. In the late 1930s and early 1940s, researchers including Landsteiner himself, working alongside Alexander Wiener, discovered a protein on the surface of red blood cells that most humans carry. It had first been studied in relation to rhesus macaque monkeys. People who had this protein were called Rh positive. People who lacked it were called Rh negative. What seemed at first like a minor footnote in blood science turned out to be profoundly important and profoundly strange from an evolutionary standpoint. Here is the strangest part. Being Rh negative means your red blood cells are missing a specific protein. At the genetic level, this comes from a deletion, an entire segment of DNA that is simply not there. This deletion creates a potentially serious medical problem. When an Rh negative mother carries an Rh positive baby, her immune system can recognize the baby's blood as foreign and mount an attack against it. Before modern medicine developed a treatment for this condition, it killed a significant number of infants. Sometimes it killed multiple children from the same mother, one after another. From an evolutionary perspective, a trait that kills your children should be rapidly eliminated. Natural selection is supposed to be ruthless about this kind of thing. A characteristic that reduces the number of offspring who survive should gradually disappear from the population over many generations. And yet, Rh negative blood did not disappear. It survived. In one specific corner of the world, it did not just survive. It reached frequencies found nowhere else on Earth. The highest concentrations of Rh negative blood in the world are found among the Basque people of the Pyrenees Mountains. The Basque Rh negative rate is extraordinary, even by European standards. And European rates are already dramatically higher than those found anywhere in Asia or sub-Saharan Africa. The Basques appear to represent a kind of genetic outlier, a population that preserved something ancient in unusual concentration. The mystery of the Basques does not stop with their blood. Their language is equally anomalous. Basque, called Euskara by its speakers, belongs to no known language family. It is a linguistic island, surrounded on all sides by Indo-European languages, but sharing no ancestry with any of them. It is the oldest living language in Western Europe, and possibly the oldest in all of Europe, a survivor from a world that existed before the waves of migration that built the modern European linguistic landscape had even begun. For researchers studying ancient human populations, the Basque combination of an extraordinary linguistic anomaly and extraordinary Rh negative frequency is a double signal pointing in the same direction. The Basques appear to be, in some meaningful sense, a living relic of an earlier Europe, a population that was insulated by mountain geography from the successive waves of migration that reshaped the rest of the continent. The Neolithic farmers who arrived from Anatolia, the Bronze Age, steppe peoples who arrived from the east, and the various historical migrations that followed washed over and genetically transformed most of Europe. But the Basque mountains provided a barrier that preserved something older. The leading scientific understanding is that Rh negative blood was carried into Europe by its earliest populations, the Paleolithic hunter-gatherers who inhabited the continent during and after the last ice age. As new populations arrived and mixed with the existing Europeans, Rh negative frequency was diluted almost everywhere. In most of the continent, it became uncommon. But in geographically isolated pockets, most dramatically in the Basque Highlands, the original frequency was preserved. The mountains did what mountains often do in population genetics. They kept things in. The Basque people are, in a very real sense, carrying a message from deep prehistory. Their blood is a window into what the genetic landscape of Europe looked like before later migrations assembled the modern European population. The Rh factor is one of the clearest elements of that ancient message. Why did Rh negative survive despite its reproductive cost? That question does not have a fully settled answer. One explanation is genetic drift. In small, isolated populations, a trait can rise to high frequency through sheer statistical chance without any selective advantage required. Another more debated possibility is that Rh negative offered some hidden benefit in the ancient world, perhaps a degree of resistance to certain parasites or infectious diseases common in the environments where those early European populations lived. The evidence for this is not definitive, but it raises a thought worth sitting with. The trait may have survived not despite its costs, but because, in the specific world of the ancient Paleolithic hunter-gatherer, The diseases present, the parasites common, and the ecological conditions of that particular time and place may have offered something worth keeping, something that balanced the cost of pregnancy complications with a benefit elsewhere in the biological ledger, a benefit that may have faded as the environment changed, leaving only the cost behind, by which point the trait had already spread far enough to persist on its own. Now hold those two facts side by side and feel the full weight of what they mean together. Type B blood surged out of the nomadic world of the Central Asian steppe. It is an eastern marker carried westward by horse-riding herders across thousands of miles of open grassland. It is most common in the middle of the Eurasian continent and the corridors that connect Asia to the Indian subcontinent. Its history is one of movement, contact, expansion, and the endless mixing that comes from a way of life built entirely around mobility. Our H-negative blood is an ancient Western marker. It peaks at the far edge of Europe in a mountain population that may represent the most intact genetic remnant of Europe's Ice Age inhabitants. Its history is one of persistence and isolation, a trait that survived precisely because one population stayed put long enough to preserve it while the world around them changed. These two traits come from opposite ends of the Eurasian landmass. They belong to different peoples, different migrations, and different chapters of the human story. They should almost never appear together, and in a person with B-negative blood, they do. They coexist in one body, woven into the same genome, the product of a chain of ancestors stretching back in two completely different directions. One toward the steppe, one toward the western mountains, who eventually, somewhere across the long corridor of Eurasian history, met each other and had children together. When you look at the history of that meeting, you can see exactly where it could have happened. The steppe was never a one-directional system. Populations carrying type B pushed westward into Europe at multiple points in prehistory. The great Indo-European migrations of the Bronze Age, which carried steppe ancestry deep into the European continent, brought Eastern genetic signals into contact with the populations already living there. The centuries of the Silk Road pressed merchants, diplomats, soldiers, and travelers from both ends of the continent into the same trading cities. The expansion of the Mongol Empire created the largest contiguous land empire in history, stretching from the Pacific coast of Asia to within reach of Central Europe and moving armies, administrators, and entire displaced communities across the full span of the continent. Each of these events was a genetic mixing engine. Each pressed together populations that had evolved apart. Across those thousands of years of contact and movement between East and West, a family line that already carried Rh-negative blood from its ancient European ancestry eventually mixed with a family line carrying type B from its steppe heritage. The result was B-negative. That combination was passed forward through whatever children survived and whatever generations followed until it arrived, rare and improbable, encoded in the cells of the body of the person reading this. This is what modern DNA analysis has made impossible to deny and impossible to simplify. When geneticists study the ancestry of populations across Eurasia today, they do not find neat, separated groups. They find a mixture everywhere. They find genomes, stitched together from multiple ancient sources that mixed and remixed across the millennia. The old idea of pure, isolated peoples with clean, separate origins has collapsed under the weight of the genetic evidence. What the DNA shows instead is a vast, tangled, continuous web of human connection, a species that has been in motion and in contact since the very beginning. This is also why B-negative people who take modern ancestry DNA tests so frequently encounter surprises in their results. Someone who identifies entirely with one region discovers threads of ancestry reaching somewhere unexpected. A family with deep roots in East Asia finds a trace of something Western in the data. A European lineage shows connections running back toward Central Asia. The blood type was hinting at it all along. The B was pointing east, the negative was pointing west, and the detailed ancestry test simply confirms, in far greater resolution, what those two small symbols were quietly encoding for generations. You're not from one place. Nobody is. But a person who carries B-negative blood carries particularly direct evidence of that truth. Evidence from both directions simultaneously, readable in their own cells and traceable across the full span of the Eurasian continent. There's one more layer to this story, and it is where genetics collides with culture in a way that is genuinely difficult to dismiss entirely. Even if the science around it remains unsettled, in Japan and South Korea, blood type is taken seriously in ways that most Western observers find surprising. Many people believe that blood type shapes personality in measurable ways, a system called Ketsuekigata in Japanese. Type A is associated with careful, responsible, precise personalities. Type O is linked to confidence and social ease. Type B carries its own distinctive reputation, independent, unpredictable, passionate, and resistant to following the crowd. In some social contexts, being type B in Japan has historically carried a mild stigma, a reputation for being difficult to work with, unwilling to conform, and hard to pin down. The scientific evidence for a genuine link between blood type and personality is essentially non-existent. Researchers have studied the claim carefully, and the data does not support it. Blood type is determined by which proteins appear on the surface of your red blood cells. There is no plausible biological mechanism by which that determination would shape how a person thinks or behaves. And yet step back from the science for a moment and notice the poetry of what is sitting there. The blood type that supposedly makes you independent and unwilling to follow the crowd is the exact blood type that came from people who made an entire civilization out of refusing to settle down. The nomads of the Central Asian steppe did not build cities. They did not recognize borders. They lived in motion, answering to the rhythm of the seasons and the movement of their animals, rather than the walls of any fixed community. They were, in the most literal historical sense, the people who would not stay in one place. The cultural stereotype and the deep genetic history rhyme in a way that is almost too clean to be accidental. It is almost certainly a coincidence. The stereotype developed long after any living memory of steppe migrations had faded. Nobody in modern Japan consciously modeled the type B personality profile on ancient Central Asian herders, but the resonance is there. The wanderers of the steppe left their mark not only in the blood of their descendants, but somehow, inexplicably, in the stories those descendants tell about that blood thousands of years later. For the people who carry B-negative blood today, all of this is more than history. In emergency medical situations, B-negative is categorically different from more common types. Finding a compatible donor can be genuinely difficult, not merely inconvenient, but difficult in ways that carry real consequences. Hospitals in Neo-Sikanwara, regions with large East Asian populations, maintain special registries for Rh-negative donors because the need can outpace the supply so severely. Rare blood networks operate internationally, coordinating donations across borders in critical cases. A bi-negative individual who donates blood is contributing something genuinely uncommon to a system that perpetually needs more of it. The rarity that makes B-negative so historically and scientifically remarkable is the same rarity that makes it medically significant. Both facts trace back to the same origin, two ancient lineages that should not have found each other, that spent most of human history on opposite sides of an enormous continent, that finally converged through the slow accumulation of contacts across thousands of years and produced a genetic combination that carries the evidence of both journeys simultaneously, 2 letters and a minus sign, on a hospital chart, one pointing toward frozen grasslands and mounted herders in the long corridors of the Silk Road, one pointing toward mountain refuges at the edge of Europe and the Ice Age populations who sheltered there while the world outside changed beyond recognition. Both ancient, both rare, both telling a story that predates every civilization whose name we still recognize, every empire whose ruins we still study, and every language whose words we can still read. If you carry B-negative blood, your blood is not a medical footnote. It is a document written, not in words, but in the language of inherited biology, recording a journey that took tens of thousands of years, crossed the full breadth of the largest landmass on Earth, and somehow arrived against all statistical probability in the specific combination that flows through your veins today. The 2 letters on your donor card are not classification. They are a summary, a compressed record of one of the longest and most improbable journeys in human history. The nomad and the ancient European, the East and the West, the steppe highway and the mountain refuge, meeting finally in a single drop of blood. If your blood type has ever surprised you, or if a DNA test showed ancestry you never expected, share it in the comments and subscribe for more documentaries uncovering the ancient stories encoded in our DNA.